This session is on infection and response.
So the key rules for this session, make sure you've printed out the booklet from the link below the video. If you've got the booklet, you don't need it in stage 1, but you will need it in stage 2 and stage 3. So, what is spatial learning science? So the plan is, in stage 1, I'll talk, you'll listen and watch. You don't write anything down, you just have to listen. You're then going to go and take a 5-minute break. You're then going to go back to the start of the video and you're going to repeat it. Once you repeat it, I'm going to talk, but you're going to do simple tasks. You're going to fill in the gaps and complete simple calculations. You'll then take another 5-minute break, and then in stage 3, you'll complete harder tasks. Now, the answers will be available at the end, but these are sorts more like the questions that you'll face in the exam papers. So we're slowly making it more complex, from you just listening, to you filling in the gaps, to you answering questions. So here we go, stage 1. I talk, you listen. Put your pens down, eyes to the front, watch the video. Here we go.
Communicable diseases can be caused by viruses, bacteria, protist, and fungi. Microorganisms that cause disease are called pathogens. Pathogens, the microorganisms that cause infectious disease, are spread by direct contact, water, or by air. Bacteria can produce toxins which damage tissues and make us feel ill. Viruses live and reproduce inside cells, which causes cell damage, but both rapidly reproduce inside the body. You need to know specifically about 3 different viral diseases. You need to know their names, as well as their symptoms and their potential treatments. The first one we're going to look at is measles. Measles causes a fever and a red rash. These are the symptoms. To prevent measles, you can get a vaccination, commonly called the MMR vaccination, which stands for measles, mumps, and rubella. If someone did not have the vaccination but gets measles, they can spread it to other people through sneezes and coughs. This is because the virus is in the droplets that they sneeze and cough out. The next viral disease you need to know is HIV. The symptoms are a flu-like illness for the first 2 weeks of infection. Unfortunately, after this point, people may not display any symptoms, so many people are unaware they are still being affected by HIV. To prevent people from getting HIV, they may take antiretroviral drugs. These drugs prevent the virus from being able to replicate. A problem of HIV is that it can progress into AIDS. AIDS is the autoimmune deficiency syndrome, which means all of the white blood cells, or the majority of white blood cells in the body, have been attacked by the HIV virus. If a person doesn't have enough white blood cells, they are not going to be able to fight disease effectively. So they are at risk of getting many other diseases or getting diseases more severely. HIV is spread by sexual contact or contaminated fluids, e.g. the passing of dirty needles between people. The third viral disease you need to know about affects plants. It is the tobacco mosaic virus, or TMV. The symptoms are mosaic discoloration of leaves in plants. So the leaves start having this yellow and green pattern. This means the chlorophyll in the leaves isn't functional. Without chlorophyll, a plant cannot photosynthesize. If there is no photosynthesis, glucose is not produced. If there is no glucose, it cannot be combined with nitrate ions to create proteins, and if there's no proteins, there's no growth. So TMV prevents growth in plants, but you do need to know the full flow of changes that happen to prevent this growth from happening. You also need to know about two bacterial diseases. The first bacterial disease we'll discuss is Salmonella. Salmonella is the scientific name for food poisoning. The symptoms of Salmonella are fever, abdominal cramps, vomiting, and diarrhea. It's spread by bacteria that have been ingested or eaten on food. The other bacterial disease you need to know about is gonorrhea. Gonorrhea is a sexually transmitted disease. The symptoms are a thick yellow-green discharge from the penis or vagina, or pain while urinating. One of the problems with gonorrhea is that many of the new strains are resistant to antibiotics. This means that antibiotics cannot be used to kill them. Instead, to prevent the catching of gonorrhea, people need to use protection such as condoms. There is only one fungal disease that you need to know about. It's a disease that affects plants, and this time it's called rose black spot. As the name suggests, the symptoms include purple or black spots on the leaves of plants. Similarly to tobacco mosaic virus, because there are black spots on the plants, the chlorophyll is non-functional. This means there is less photosynthesis, and obviously the plant will not be able to grow. Other problems rose black spot can cause are the leaves falling off. This is dangerous because the leaves will contain the fungal spores, and if they transfer to another plant, that plant will also become infected. The fungus can also spread through the wind, so prevention or a cure is treating the plant with fungicides or removing the affected leaves and burning them. Remember, anything that ends with -ides is going to kill something. So fungicides kill fungi, pesticides kill pests, insecticides kill insects. One last disease you need to know about is from a protist. A protist is a eukaryotic organism, and in this case it is causing the disease malaria. The main symptom of malaria is fever, but one of the main problems is that it can be fatal. Malaria is spread by mosquitoes, which are vectors. A vector is an organism which transmits disease but does not get the disease itself. To prevent people from getting malaria, the most common prevention method is using a mosquito net, or methods to avoid being bitten. So these could be using things like insecticides to kill the mosquitoes, or preventing them from breeding by removing stagnant water. The human defence system. Humans have a non-specific defence system to destroy pathogens, or to prevent pathogens from entering the body. The skin is the largest physical barrier. It blocks pathogens from getting to where they want to go, which is inside your body. In your respiratory tract, so the trachea and the bronchi, there is mucus to trap pathogens. In the stomach, there is acid, and a common misconception is that this acid helps to digest food. It does not. It is only there to kill pathogens. If pathogens manage to evade these initial defense tactics, your body has white blood cells to help to destroy them once they're actually inside the body. Step 1 is phagocytosis. This is a non-specific response. It does not matter whether you've seen this pathogen before, whether it is a virus, a protist, a fungi, or a bacteria. The white blood cells will recognise it, engulf it, and then digest it using enzymes. The next two responses, making antibodies and making antitoxins, are really specific responses. Antibodies are made against a particular antigen on a pathogen. So an antibody against salmonella would not work against gonorrhea. Antitoxins are specific to bacteria, and they are specific to the toxin the bacteria has made. This diagram represents phagocytosis. In the top right-hand corner, you can see the white blood cell is engulfing the pathogen. Once the pathogen is completely engulfed and inside the white blood cell, enzymes will bind with it and cause it to become digested. Once it's been broken down into non-harmful components, these can either be excreted from the white blood cell or they can be retained for future use. As we said before, antibodies are specific to the pathogen that is infecting the body. Antibodies bind to the antigens of pathogens. There are so many anti-words in this topic, but to remember antigens come from pathogens, look at the ending of the word. Both words end in G-E-N-S, so you know antigens are on the pathogens. Once an antibody has bound to the pathogen, it either targets it for destruction or it might bind to another pathogen, causing them to clump together into an easy to clean up area. Vaccinations. Vaccinations help prevent infection in living organisms. A vaccination contains a small amount of a dead or inactive form of a pathogen. These pathogens still have antigens on their surface, so the white blood cells will still be stimulated to produce antibodies. This means if the same pathogen re-enters the body, the white blood cells can respond more rapidly to produce the correct antibodies. This will prevent infection from happening. It's so important when you write these answers that you mention upon secondary infection, the production of antibodies is faster. That is the key difference between primary and secondary infection. You need to know about antibiotics. Antibiotics, such as penicillin, are medicines that help to cure bacterial diseases by killing the infected bacteria inside the body. It's important that specific bacteria can be treated by specific antibiotics. So some antibiotics would be better at treating gonorrhea, others might be better at treating salmonella. The use of antibiotics has greatly reduced death from infectious bacterial diseases. However, the emergence of strains of resistance to antibiotics is of great concern. Really importantly, you must remember antibiotics cannot kill a virus. Remember, viruses replicate inside your cells, so they are protected from antibiotics because they cannot get into the cell. Another form of treatment are called painkillers. Painkillers and some other medicines can be used to treat symptoms of a disease, but they do not cure the disease because they cannot kill pathogens. This is completely different to antibiotics, which do kill bacteria. If you just take painkillers, you are making yourself feel better by masking the pain, but you aren't curing the initial cause of the disease. Killing viruses is incredibly hard because of their nature, they are replicating inside your cells, which means in order to get to them, you must damage your body's own tissue. Developing new drugs. We can get lots of different drugs from plants. You need to know about 3 specific examples of drugs and their origins. The heart drug digitalis originates from foxgloves, the first image on the left. The painkiller aspirin originates from the willow tree, and penicillin was discovered by Alexander Fleming from the Penicillium mold. Drugs go through a really extensive testing process to make sure that they are safe for human consumption. There are 3 specific things you need to know that drugs are tested for. The toxicity is whether or not they have negative side effects. The efficacy is whether they do the job that they are intended to do. For example, if a heart drug is attempting to bring down the pressure in the heart, is it doing that, or is it simply making the heart beat more slowly? And the dosage. What is the minimum amount of drug required to have the desired effect? When drugs are tested, they go through 2 different stages, preclinical and clinical. Clinical means human trials, so preclinical means before human trials. The order is test on cells, test on tissues, test on live animals, low doses given to healthy volunteers, low doses to unhealthy volunteers, then large doses to unhealthy volunteers. Remember, all throughout these tests, patients and doctors will be monitoring efficacy, dosage, and toxicity. To test the effectiveness of drugs, volunteers can do a double-blind trial. This is where there are 2 categories of patients. One set of patients will receive the active drug. The other set of patients will receive an identical formulation which does not contain the active drug, also known as a placebo. In the double-blind trial, neither the patients nor the doctor know who is getting the active drug. The placebo will have no effect on the condition, but will help to show a baseline and act as a control group.
Right, that's the end of stage 1. Now all you need to do now is you need to go and take a break for 5 minutes and forget about that information. Then when you come back to start stage 2, I want you to skip to the start of the video and replay the first part of the lecture. Now, as you listen to it this time, you're going to complete the close activity. You're going to fill in the gaps on the booklet for stage 2. So make sure you've got that booklet and fill in the gaps for stage 2. Now, hopefully, you've just completed stage 2. That means you're now ready to go on to stage 3. Again, you need to take a break, a minimum of 5 minutes. Now, in stage 3, you're going to press pause at this point. Once you've pressed pause, you're going to answer the questions in stage 3 of the booklet, and you've got 15 minutes to do that. When you press play, the answers will then appear over the next few minutes.
So, your 4 answers for what can cause communicable diseases should be viruses, bacteria, protist, and fungi. A pathogen is a microorganism that can cause disease. Pathogens can spread through direct contact, water, and through the air. Bacteria make us feel ill specifically by producing toxins, and viruses make us feel ill by living and reproducing inside cells, which eventually causes the cells to burst, causing wide-scale damage. You need to remember which type of pathogen causes which disease. So measles is a virus, HIV is a virus, tobacco mosaic virus as it says in the name, is a virus. Salmonella and gonorrhea are both bacteria. Rose black spot is a fungi, and malaria is a protist. What disease is it? I affect plants. I cause a mosaic discoloration of leaves. I reduce photosynthesis. So what am I? I am tobacco mosaic virus. I am ingested in food. I cause vomiting, diarrhea, and abdominal cramps. I am a type of bacteria. What disease am I? I must be Salmonella. There are 3 things white blood cells can do to protect against pathogens. These are phagocytosis, producing antibodies, and producing antitoxins. Antibodies bind to the antigens on pathogens. In a vaccine, you find a dead or inactive form of the pathogen. After a vaccine, white blood cells will create antibodies. Antibiotics can only kill bacteria. They cannot kill viruses. If someone has HIV, would a doctor give them antibiotics? No. HIV is caused by a virus. Antibiotics cannot kill a virus. How are painkillers different to antibiotics? Painkillers reduce the symptoms, but they do not kill pathogens. Drugs are tested for 3 different things: toxicity, efficacy, and dose. Make sure you know what each of these things are. When we develop new drugs, they are initially tested on cells, tissues, and animals. These are the preclinical trials. They're then tested on healthy volunteers and then unhealthy volunteers. If it is safe, they can continue. If not, they stop testing. In a double-blind trial, neither the doctor nor the patient knows if they are having the drug, and a placebo is an identical formulation to the drug without the active component, so a chemical which will have no effect on the illness.